Hello, everyone, and welcome to today's webinar titled SARS-CoV-2 Neutralizing Antibody Testing for Vaccine Efficacy Assessment. This webinar is part of the Clinical Diagnostics Research event, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about our sponsor, please visit them at genscripts.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate first by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right-hand side of your screen. By submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located at the right of the abstract. This survey will be available during both the live session and the on-demand period. Now, if you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Sean Taylor, North American Field Application Scientist Manager at GenScript USA. For a complete biography of our speakers, please visit the Biography tab at the top left of your screen. Dr. Taylor, welcome. You may now begin your presentation.
Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. And now that we have reached a stage where multiple global populations have been vaccinated by a variety of different vaccines, a new approach to testing may be required in this pre- and post-vaccination era. So some critical questions surrounding the SARS-CoV-2 pandemic have arisen. And the first one is associated with the commercial vaccines themselves, which have widely different efficacies, begging the question, how am I assured protection post-vaccination in the context of the wild type and variant strains that are circulating now with varying vaccine efficacies? The second question is associated with the persistence of immunity. When I get vaccinated, how long am I actually immune before I potentially need a booster shot? And the third question is related to the first two questions: what tests are relevant and provide the most actionable information in populations of mixed vaccinated and non-vaccinated individuals? There are two major categories of testing, one being serology, which are antibody-based tests, and molecular, which are the RNA/DNA-based tests to assess the live virus circulating. So the serology tests are the signature of the virus after being either infected or vaccinated with an immune response and then antibodies that are generated within the host consequent to SARS-CoV-2 infection or vaccination. The molecular testing is really to assess the presence of live virus, the presence of active infection early on after the infection has begun. But once the immune response has taken over, the viral RNA levels go way down because the immune system has responded and attacked the virus and destroyed the virus and prevented the virus from reinfecting cells. And what we're left with are the signature of the virus from the immune response, which are the antibodies now that are circulating to SARS-CoV-2. Amongst all the antibodies that are generated in an immune response, there's a very special subclass of antibodies which are called neutralization antibodies. And these antibodies have a very defined function in blocking the virus from docking to the host cell. So in SARS-CoV-2 infection, the spike protein, the tip of the spike protein, which is called the receptor binding domain, interacts with the host cell ACE2 receptor, so the extracellular domain of what's called the ACE2 receptor in a host cell. So this would be the lipid bilayer membrane of a host cell, and this would be the ACE2 receptor. So this is the intracellular part, and this is the extracellular part, which interacts with the receptor binding domain of the spike protein of SARS-CoV-2. So it's a very strong interaction, and when the virus binds or docks to the ACE2 receptor, it can then inject its RNA into the host cell to make more viral particles. So this is what propagates infection. So if we can block the virus by the binding of antibodies to that tip of the spike protein, to the receptor binding domain, then the virus can't bind to the ACE2 receptor and therefore will not be able to replicate and propagate infection. So the virus is essentially rendered inert, unable to propagate infection. So these neutralizing antibodies are what promote or prevent the virus from actually binding to the ACE2 receptor. And as we see here with this dartboard, if the dartboard represents the spike protein, let's say, and all these darts represent antibodies that are generated by the immune response, we can see that the antibodies, none of these antibodies are hitting anywhere near the spike protein itself. There's a lot of antibodies being generated, but they're not neutralizing. As we zoom in on the dartboard, we can see, oh, in fact, that some antibodies actually are towards the spike protein. And there's one in particular, this green arrow, which is right in the bullseye, and that's a neutralizing antibody, which would bind to the receptor binding domain. So as I mentioned, in the immune response, many antibodies are made. The immune system doesn't know what and how to make a neutralizing antibody. It just makes a whole bunch of antibodies. But some of them are neutralizing, and those are the important ones because they block the virus. So ideally, we would be testing for neutralizing antibodies then in the immune response to either infection or vaccination, because those are the antibodies that really are the ones that are important in blocking and preventing infection. Now, there are some pre-existing neutralization tests that are out there that test for neutralizing antibodies, and both of them require either live SARS-CoV-2 virus or a pseudovirus that expresses the spike protein of SARS-CoV-2. So this is a safer test because this is just an innocuous virus that makes just the spike protein. So we can use this in a BSL-2 lab. However, if we're using a virus neutralization test with live virus, then this is very dangerous, obviously. We don't want to get infected with SARS-CoV-2, so it requires biosafety level 3 laboratory containment facility, which is they're not that common, and they also require very special training and equipment to work in that kind of an environment. Both of these types of tests require a significant amount of time to run the test to assess neutralizing antibodies in a patient sample, so in a blood sample. Typically 2 to 4 days of work using live cells and live cell culture and live virus and culturing virus, which is all very time consuming and requires specialized training. So GenScript released a novel assay that uses not live virus or live cells, but the protein components of the virus and cell that interact with each other. So as I mentioned, the interacting components are the receptor binding domain of the spike protein of the virus. So the very tip of the spike protein is the receptor binding domain, and the extracellular domain of the ACE2 receptor depicted here in pink. So we teased out these protein components and expressed them and purified them so that we have in green here the ACE2, the extracellular domain of the ACE2 receptor and the receptor binding domain of the virus. And we tested these two proteins to confirm, in fact, that even when separated, when taken, when expressed individually, that they do interact very strongly. So they complex together very strongly. So they mimic this interaction between the live virus and the host cell. So by coating ELISA plates with the ACE2 receptor, as we've done here, then all we need to do is incubate the receptor binding domain depicted here, this protein, with patient samples. And if those samples contain neutralizing antibodies, as we see here depicted in red, then those neutralizing antibodies will bind very strongly to the receptor binding domain protein and block it from binding to the ACE2 receptor. There will be no color change in the assay. However, if an individual doesn't have neutralizing antibodies, so you see here the red ones are neutralizing, but the blue ones, they still bind somewhere on the receptor binding domain, but they're non-neutralizing, they're non-functional, so they permit the receptor binding domain to interact with the ACE2 receptor, as we see here. And if this interaction happens, then the color of the solution turns yellow. And the darker the color, the less neutralizing antibodies are in the patient sample. The advantage of this test, apart from the fact that it's now an ELISA test with protein components that permits this to be very high throughput, much more high throughput than any of the live virus tests that are out there, and much less expensive than those tests as well, is that it's immunoglobulin agnostic as well. So this test doesn't have any specificity for the subclasses of immunoglobulins like IgA, IgG, IgM, etc. Any immunoglobulin that has a neutralizing antibody component to it will bind to the receptor binding domain and block it from binding to the ACE2 receptor, and will measure neutralization. So it's a very sensitive test because it's not limited to a specific immunoglobulin. The second advantage is that the receptor binding domain is in solution. And this is a big advantage because this assures that the receptor binding domain is in a near native conformation, which assures that the receptor binding domain antigenic sites are very close to those that would be found in the live virus, forcing the antibodies that are generated to bind very specifically to the receptor binding domain. And the third key advantage to this test, of course, is that it is a functional assay. It only measures the functional neutralizing antibodies that block the interaction between SARS-CoV-2 and the ACE2 receptor, which are the important antibodies required for protection. We did do a recent study this year, 2021 actually, which we published in Journal of Clinical Microbiology, that compared our CPAS surrogate virus neutralization test to other binding antibody tests. And when I say binding antibody tests, I mean tests that do not delineate between neutralizing antibodies and all the other antibodies that are made in an immune response, which simply bind somewhere to the virus but may not be neutralizing. And in those comparisons, we found that our CPAS test was very comparable in terms of specificity and sensitivity, either equal or had much higher specificity or sensitivity to the other commercial tests that were used in this study. And you can review this study to learn more information on this. We also compared the test to the gold standard live virus tests. And what we found was that there was excellent correlation to those tests, upwards of 98 to 99% correlation. Another study compared the SARS-CoV-2 surrogate virus test, or CPAS test, to live virus tests, again, in a variety of samples. So you can use it to look at neutralizing antibodies generated in any type of organism, so long as it was infected with SARS-CoV-2, to assess the neutralizing antibody response. And in fact, in this study, there was over 400 human samples, and the correlation between our CPAS surrogate virus test and live virus, PRNT testing, which is plaque-reducing neutralization test, which is one of the gold standards of the gold standard live cell tests, was approximately 99% sensitivity and specificity. Now, we have released a new protocol for semi-quantitative analysis of the neutralization antibodies generated from infection or vaccination. This is only for research use only. The kit has been FDA authorized for qualitative analysis to assess positive or negative samples for neutralization antibodies. This is to assess the levels of neutralizing antibodies between samples. I'm not going to go into the details of the actual protocol, but suffice to say that it requires generating a standard curve from a standard antibody that we provide that can be purchased separately with the kit. So this is to produce the standard curve. And then you can plot on that standard curve the OD of a given sample to assess the titer of the sample. So if we look at all the different vaccines that are out there that are commercialized, and there are many other ones since this was published, but just to give an example, the efficacy versus the original strain, the original SARS-CoV-2 strain that initiated the pandemic way back in 2019, Pfizer was 95% effective and the Moderna was 94% effective. They never were able at that time to test their vaccines against the variants because the variants didn't exist at that stage. So all the data we had were these results. When the Johnson & Johnson vaccine came out, though, the South African variant was actually prevalent, and they did do some clinical trials in South Africa to assess efficacy. And we can see that against the original strain, it's 72%, which is much lower actually than the Moderna and Pfizer, but even lower at 57% in the presence of the South African strain of the virus. So this is concerning, obviously, for someone who might receive this vaccine. Same thing for the AstraZeneca vaccine, where you can see that the overall efficacy is 70% against the original strain. But in South Africa, there was only a 10% efficacy, and they actually stopped using the AstraZeneca vaccine as a consequence of this. It was as if no one was actually getting any protection from vaccination. If we go down the list further, we can see that the Sinovac vaccine only has 50% efficacy to the original strain. So if we look at all these data combined, again, it begs the question, if I receive a certain vaccine, am I really protected post-vaccination? More real-world efficacy data, which is a little bit concerning, is the elderly and immunocompromised individuals. And these two studies here are, one was a study in Germany performed on people that were under 60 and over 80 years old. And what we see here is if we look at the negative cutoff from the serology test that they did, what they found was that many of the people that were over 80 years old were falling in this not generating any significant antibodies post full vaccination. And this study used the Pfizer vaccine, and these were all people who were fully vaccinated, where they determined that about 30% of people over over 80 years of age don't generate measurable levels of neutralizing antibodies. In another study, which was performed in Canada, in Toronto, this was looking at immunocompromised patients who were on immunosuppressive drugs after solid organ transplant. And what they found in this study was that over 70% of immunocompromised patients receiving the Moderna vaccine did not elicit a neutralizing antibody response. Again, this is very concerning. It's not very surprising because they were on immunosuppressive therapy. So the immune system is already compromised when they receive the vaccine, but still very concerning that such a large proportion of people who are on immunosuppressive therapy are not having a robust neutralizing antibody response after vaccination. 3 major studies correlate vaccine efficacy and protection. So these are very new studies that looked at vaccine efficacy versus the neutralizing antibody levels in those individuals. And what these studies all find is that there is a kind of a cutoff titer somewhere around 100 to 200 WHO international units. The vaccine efficacy seems to drop quite precipitously. And all of these studies that have been done kind of correlate each other pretty well, actually, considering the large variability in these studies. The Corey paper, also published in Nature Medicine, found that at 54 international units, protection is down to about 50%. So again, quite well correlating with each other, all of these studies. So now, if we look at the comparison between binding assays, so remember, binding assays look at all the antibodies that have been generated by an immune response to a particular antigen, like receptor binding domain or nucleocapsid protein or full spike protein. And if we compare those assays to the neutralizing antibody test, the CPAS test that we've released, what we found was that for two of these tests, which are just looking at IgG binding, that after using the Pfizer vaccine, after 2 weeks post the first injection of the Pfizer vaccine, it looks like all the patients, almost 100% of them, have a strong antibody response. But if you compare those two immunoglobulin binding tests to our neutralization test, which is this bar in dark blue, we see a significant difference. Much less people in the study group have neutralizing antibodies in comparison to the people that have binding antibodies. And with the Moderna vaccine, looking at a more elderly population, of course the results are lower as expected because elderly people tend to have a less robust immune response to begin with. But that said, if you compare the immunoglobulin binding antibody test to the neutralization test, you see a marked decrease in the number of people that have neutralizing antibodies versus binding antibodies. So it begs the question, should we be more careful about the testing we do and measure neutralizing antibodies to assure which individuals have protective neutralizing antibodies as opposed to knowing that people just have binding antibodies? This article where they looked at 4 individuals and found that after the first dose of vaccine, which is here, leading up to the second dose, there was a very low level of neutralizing antibody titers. But right at the second dose, which is in this gray zone here, when all these people got their second dose, all these lines go up, as you see in titers. So very robust response in immunity after the second dose, which is good. So lots of neutralizing antibodies. But then we measured their response after the second dose and saw that the titers dropped in some cases quite precipitously. So for patient 156, in other cases a little more gradually. So it really does depend on individual response how these titers drop. And we still don't know what level of titer can be a good predictor of when to get a booster shot. But for sure, the immune response in terms of neutralizing antibody levels is decreasing significantly after the second dose, as we see here. If we look at the case for Israel, which was one of the first countries to actually receive a vaccination from the Pfizer vaccine, we know in their case that they entered a 4th wave of cases in August. And that wave was as big as any of their other waves. And this was after their population had been almost fully vaccinated with the Pfizer vaccine. So there's definitely an issue of waning immunity over time, and particularly in the presence of the variant. So most of their cases were from the Delta variant in this case. And they initiated a booster program, and now their caseload is dropping down again almost to 0. The UK is now very much still in their 4th wave, with, again, highly vaccinated population pretty early on. And again, this suggests that the immunity from that AstraZeneca vaccine is waning to the point where people are now more susceptible to the variant strains that are circulating in the UK. And the question is there a way now to be more precise about measuring this, like when people would need a booster shot as opposed to just determining a random time frame for people to be boosted. So in conclusion, PCR testing only represents a moment in time. When we get tested with a PCR test, we only know that we're negative at the moment that we took the test. It doesn't mean that we are assured of protection at any time after being tested. We could easily be infected at any moment after a PCR test. The presence of binding antibodies also does not mean that functional neutralizing antibodies are present. We've learned this from the previous study here, where we see that you can have a lot of binding antibodies, but no neutralizing antibodies. Furthermore, a neutralizing antibody can persist for months and possibly longer, as we saw from here. These are days after infection, so after several months, neutralizing antibodies are still circulating in these patients. So a positive neutralizing antibody test can indicate a persistent level of frontline functional antibodies that can potentially block the virus from propagating and docking and entering cells and propagating infection. So given that vaccine efficacy varies widely, a CPAS test may help to assure that the expected response to vaccination was elicited. And neutralizing antibodies can also be measured longitudinally, as we saw here. We can measure them over time to assess how the antibody levels are waning and whether protection is still assured. So therefore, for mixed vaccinated and unvaccinated populations, a combined approach may be the best way to go about testing now in this post-vaccination era, where we use PCR for those who don't have immunity or have not been vaccinated. And we can use CPAS, or neutralizing antibody tests, for vaccinated individuals or people who may have been previously infected to assess their levels of neutralizing antibodies. For more information, we have recently published a very short review article in the Journal of Clinical Virology Plus. It's only a 2.5-page read. It's written in very plain language, very well referenced with the latest references, and I strongly recommend this article as to put on your reading list if you'd like to learn more about anything related to this talk. And I appreciate your time and attention.